focused on plant metabolomics. And this is an old figure. So this reported number of 200,000 plant-derived natural products is much higher, and recent sources say closer to 1 million. But this is a good figure to illustrate how metabolic complexity is being generated within plant-specialized metabolism. So we're going to start from a pool of primary metabolites and all of the metabolic pathways that are feeding that, and they'll then get further modified to form scaffolds that are then decorated through glycosylation, methylation, and other enzymatic reactions. So I think that when some people think about diversity in the plant kingdom, they might think about morphology. They think about the different leaves or flower shapes and colors. But plants, as many of you know, create really diverse chemicals. And they're complex and they're useful. And all of these, so whether it's the floral shape and color or complex terpenes or alkaloids, this is driven through evolution. So you could think of the fence as being very physical, whether it's the thorns on a honey locust tree or the pricks on a cactus. But you could think about acyl sugars or the white latex sap of the poinsettia plant you buy at Christmas as defenses as well. You could also think about floral volatiles or the phytochemicals that you might find in fruit or nectar to attract pollinators as another way that plants are using these phytochemicals. And I think that what's important to note is that both, whether it's defense or pollinator attraction, that these are all going towards reproductive success and survival. So all of this to say that plants are master chemists, and humans have utilized these metabolites for thousands of years. The metabolites that I'm showing you are bioactive, and some may be recreational drugs if you're thinking about precursor to MDMA or cocaine, but they're also really important medicines. And so sort of Plaxitaxel and artemisian and scopolamine are success stories of medicines within plants. Scopolamine and cocaine are both tropane alkaloids, and that's important because our lab focuses on tropane alkaloids in the Solanaceae, so the tomato family. And Atropa belladonna is a scopolamine producer that has a rich history as a poison. And you can find numerous case studies about people accidentally eating berries and becoming really sick. But the plant also produces two very medicinally important compounds, scopolamine and hyoscyamine or atropine. So if you've ever seen a scopolamine patch, it's used to treat motion sickness, someone might wear it. And that's a great example of a use of this essential medicine. So like many plant-specialized metabolic pathways, the production of tropane alkaloids is phylogenetically restricted. So as I mentioned, the scopolamine pathway is found in Solanaceous plants, so Datura, Hyoscyamus, and Atropa. But you can find tropane alkaloids in a few other plant families. So you can find them in Moraceae and mulberry. You can find cocaine, which is a different tropane alkaloid, in Erythroloxaceae, so coca, and you can also find them in the Brassicaceae and Cochlearia. This is the tropane alkaloid biosynthetic pathway as it is characterized. And there are three main entry points into this pathway. The first being putrescine, which leads to the production of the first ring of the tropane core. So putrescine goes to the generation of the N-methylpyrrolidine cation, which is a pyrrolidine. You also require malonyl-CoA to form 3-oxoglutaric acid, and that's also going to generate the tropane core. To generate tropane aromatic esters, so when I refer to tropane aromatic esters, I'll be talking about tropane esters with other aromatic acids attached through an ester bond. You need to form phenolactic acid. So the first tropane aromatic ester in scopolamine biosynthesis is tropene esterified to phenolactate. And that requires the funneling of phenylalanine into the pathway. So I'm going to talk about these genes later on. And so it's important to note that phenylalanine is modified into phenylpyruvic acid through an aromatic amino acid transferase. And then that is reduced to form phenolactate through phenylpyruvic acid reductase. And our lab is focused on discovering chemical diversity in alkaloids outside of this pathway. So as I mentioned, the formation of the N-methylpyrrolidine cation is important to tropane alkaloid biosynthesis because it makes up the first ring of the tropane core. But this pathway, or the production of the N-methylpyrrolidine cation, is conserved in the Solanaceae. So you also find this in nicotine biosynthesis. And in fact, there's a lot of diversity in pyrrolidine alkaloids, and this is across kingdoms. So you can find different decorations of pyrrolidines, whether that's hydroxylations or the addition of aliphatic chains or aromatic moieties. To characterize diversity outside of the pathway, we first silence the polyhedride synthase responsible for the reduction of 3-oxoglutaric acid. When we do this, we get an expected increase in the N-methylpyrrolidine cation and a decrease in tropane alkaloids. I'd like you to pay attention to the increase in this molecule because that's quite a lot. We then use discovery metabolomics to uncover the metabolites that differentiate the polyhedride synthase plants from our infiltrated control plants. And so we perform VIGs, we use our QTOF to do LCMS profiling, and then peak alignment and picking leads to data filtering, so removing contaminants, and then we can then do multivariate statistical analysis. And so the first plot that I'm showing you is an OPLSDA analysis. So that is going to examine the differences in the LCMS profiles of our two genotypes. And when we do this, we get a really clear separation of our PYKS silenced compared to our TRV2 control plants. And this is the loadings or the S-plot of the OPLSDA analysis. And each individual dot on this graph represents an individual metabolite feature, where those that are further from the x-axis are contributing most to the difference between these two genotypes, and those further from the y-axis are the most abundant. We can then go and do tandem mass spectrometry on each of these metabolites, or those that we're able to perform tandem mass spectrometry on, and make assignments of class. And so, as you would expect, we can assign tropanes based off of metabolites that fragment to give tropane core-like fragments, so a tropanone or a tropene fragment. And they were all decreased. So all of the tropane alkaloids that we were able to get tandem mass spectrometry annotations for were decreased in polyhedride synthase silence plants compared to control. So here, this heat map is showing log-fold change in polyhedride synthase plants compared to TRV2. We can then go in and annotate pyrrolidine alkaloids. So those are the compounds that are fragmenting to give 84.08, or the N-methylpyrrolidine cation. We looked closely at the tropanes that were decreased in our polyheptide synthase plants. And there were many that were known. So littering, hyoscyamine, 6-hydroxyhyoscyamine, and scopolamine are all known metabolites in the scopolamine biosynthetic pathway. But there were many that weren't known and were novel. So that includes the addition of phenolactoyl moieties, glycosylations, malinations, and a common theme among this was that many pathway metabolites were being decorated to increase metabolic diversity in tropane alkaloids. But for this story, we're mainly focused on pyrrolidine alkaloids. So those are alkaloids that are fragmenting to give 84.08. And when we look at those compounds that I've annotated as pyrrolidines, they're increased except for P3, and P3 is high green. So high green requires the production of 3-oxoglutaric acid. And so when you silence polyheptide synthase, you're getting an increase in the N-methylpyrrolidine cation, but you're not getting 3-oxoglutaric acid. So you can't form high green. We then wanted to focus on two of the most abundant pyrrolidine alkaloids, mainly because you have a better chance of getting a confident metabolite ID, and you have a better chance of being able to purify those metabolites and perform NMR. So they're the two most abundant pyrrolidine alkaloids in our analysis were isomers of 292. And our annotation, our structural annotation for that compound looks like this. And I'm showing the tandem mass spectrometry here. It first fragments to give N-methylpyrrolidine to acetic acid, so this 144 fragment. And then it gives the N-methylpyrrolidine cation, so the 84.08. We wanted to test whether the aromatic acid portion of this molecule was being formed through the formation of phenolactic acid. And so in order to test that hypothesis, we silenced the phenylpyruvic acid reductase responsible for the production of PLA in Atropa belladonna. And so when we silence the PPAR, we get an expected decrease in phenolactic acid and malonyl phenolactic acid. So that's this metabolite. And we also get a decrease in both isomers of our unknown, 292. We wanted to look at the formation of these compounds in detail. And so we did this in two different ways. The first being transient expression in Nicotiana benthamiana. And so we transient express the genes that are responsible for the production of phenolactic acid and for the production of the N-methylpyrrolidine cation. And when we do this, there's an endogenous Nicotiana benthamiana malonyl transferase that malinates phenolactic acid. And so you get the production of 2O-malonyl phenolactate, or MAL-PLA, and the N-methylpyrrolidine cation. These can then combine to form both isomers of the 292 compound. So here I'm showing a polyheptide synthase route, and in blue, the blue chromatogram is showing the production of both isomers of 292 and their co-retention with our Atropa belladonna root sample. We then wanted to test if these reactions could occur in the absence of enzyme. And so to do that, we purified 2O-malonyl phenolactate from belladonna. We chemically synthesized the N-methylpyrrolidine cation. And when we combine these two in the absence of enzyme, we get the formation of both 292 and the dipyrrolidine version of the 292 molecule, so 375. And so in orange, I'm showing our in vitro reaction, and that also co-retains with our PYKS silenced Atropa belladonna root samples. And then I'm showing the production of the 375 isomers. So here I'm proposing a mechanism for this non-enzymatic reaction. And it really resembles a managed-like decarboxylative condensation, where 2-malonyl phenolactate is enolizing to form a nucleophile that's going to attack the electrophilic N-methylpyrrolidine cation. You lose this as CO2, and then you form the 292 compounds. We were then able to bulk silence PYKS and harvest all of those roots to get material for purification of the 292. So on the top, I'm showing you a chromatogram of polyhedride synthase silenced plants, and it's enriched in pyrrolidine alkaloids, these two being our two main 292 peaks. And then you can see the dipyrrolidine version here. We're able to purify that through semi-preparative fractionation and get a sample that contained both 292 isomers, and then further purify that using an isocratic method on the semi-preparative system. So here I wanted to remind you that I brought up these 375 compounds, which are quite abundant, but I didn't talk about them in detail. And after annotating and validating the structures of the 292, we wanted to understand more about pyrrolidine diversity in Atropa belladonna. And so we looked in more detail at the different pyrrolidines and their fragmentation. So on the bottom, I'm showing you the 292 sort of as a reference. So you'll see how this compound fragments. And here I'm showing you the dipyrrolidine fragmentation. So it fragments to lose methylamine to this fragment. You can then lose phenolactate and only retain this portion of the molecule. And then it's giving that typical pyrrolidine fragment. Here we have a hexose addition to 292, where you get a 292 fragment from this molecule, and then it fragments just like the 292 molecule does. So after diving deeper into our increased molecules in the PYKS silence plants, we saw that many of them were simple decorations of the 292 compound. So you can see the addition of hexoses, of pentoses, of phenolactoyl moieties. We see simple hydroxylations, hydrolysis, and we see similar things with the dipyrrolidine version. And I think what's important to note is that here we're able to report alternative fates of pathway intermediates. So the N-methylpyrrolidine cation and phenolactate are both required for scopolamine biosynthesis. But here we're showing that they can also go into form a whole suite of this class of alkaloids. This novel compounds at that. So pyrrolidine diversity is generated both through non-enzymatic reactions, where that comes to the 292 and the 375, but also promiscuous enzymatic activity. So hydroxylations, glycosylations, which in specialized metabolism are notoriously promiscuous. So I wanted to remind you of this mechanism and also remind you that many of the compounds in our multivariate statistics were left unknown, sort of this metabolomic dark matter. And we wanted to take a reverse metabolomics approach to elucidate their structures and annotate these compounds. And so in order to do that, we wanted to take advantage of the electrophilicity of this cation and combine it with molecules that are known to be in plants. And so we came up with a list of potential nucleophiles that we thought would be reactive with the N-methylpyrrolidine cation. So that includes anthocyanins, aromatic acids, flavonoids, different alpha keto acids, and we combine these with the N-methylpyrrolidine cation in vitro. And when we do this, we get all of this metabolic complexity. We get flavonoid alkaloids, which have been reported in ficus, so that's a plant in Moraceae, and you can see that they're able to make multiple additions. So that's quite crazy, I thought. But unfortunately, the reverse metabolomics approach wasn't very fruitful. And I think this is mostly owed to the many unknowns in a nucleophilic substrate pool. So you could think that the acids that are available in the root and the leaf are very different from each other. And also the complexity in plant enzymes makes it hard to find the acids that might be commercially available. So many of the acids that are in plants can then get further modified through glycosylation or myelination, making them unavailable to us. So with that, I'd like to talk about some of the implications of the research. The first being that there's a lot of interest in engineering this pathway. And you could think about the electrophilicity of the N-methylpyrrolidine cation as sort of inefficient. If you have this metabolite combining with other intermediates in the pathway to form these products that you might not want, you can think of that as an inefficiency for engineering. Also, it's novel to think about the role of non-enzymatic reactions in metabolism. So I'd like to think that biochemists are quite biased in their thinking about there always has to be an enzyme involved, and that's not always the case. And sort of a homage to chemical evolution. So if we think about how enzymes evolve to do the functions that they do, how enzymes evolved in the history of life. If you think about non-enzymatic glycolysis and the evolution of the enzymes to form that function, enzymes are serving to lower the activation energy of reactions that can already occur. So with that, I'd like to thank our funding sources. I'd like to thank my research mentor, my collaborators, and all of the people who have made this work possible, especially my undergrads who have worked with me over the years. So I will take questions and thank the audience for their attention.
Thank you very much, Hannah. Any questions, you can feel free to put them in the chat or to unmute yourself and ask away.
Oh, Gaurav.
Hi, nice talk. I was wondering, for pulling out the diversity of the pyrrolidine and the other alkaloids, did you use that single ion, or are there coupled fragments that you can also use for being more sure about that being a specific metabolite that you're interested in?
So for pyrrolidines, we mostly focused on the 84 fragment. Though there are metabolites in the analysis that give different fragments, there are still just like many unknowns.
Right, but what about other metabolites that also produce the 84.07 fragment?
Right, so there definitely are some of the analysis. It's just we don't know what the acid portion of the molecule is, if that makes sense. And when we were doing the reverse metabolomics, we were trying to use that as a strategy to uncover what the acid portion of the molecule was. But I think that in the case of some of them, the acids were further decorated, and so they weren't commercially available to us.
There's a question in the chat here from Kirana. Kirana says, hi, nice talk. How did you silence the PPAR or PKS genes using metabolic engineering or inhibitors?
So we use virus-induced gene silencing. So that is sort of works great in our system. And I mentioned it briefly at the beginning of my talk, but I didn't go into detail. But it works through RNA degradation. So we're using a viral vector to use the host RNA silencing mechanisms to degrade mRNA.
Other questions for Hannah?
Hi, yeah, I had a question. Thank you for the talk. I was wondering about these compounds that I can't remember what it was called, but it had the mass of 375 and it had this dipyrrolinium attack that happened. And I was wondering, you're measuring the mass at 375, and I guess you're measuring in positive mode when you're doing this ionization. Do you ever see the doubly charged? Because I imagine it would be quite possible for the doubly charged to be there, in which case I don't know if I saw it on the mass spectra there. I think I only saw the 375 if you did show that. But do you ever see that doubly charged ion?
So it might be there. I haven't ever looked for it.
Okay.
But you would think that if you have like a 2+ charge, then it's going to be cut in half because it's it's over Z, the mass is cut in half. So yeah, I haven't seen it.
Okay. I was wondering if like you're doing any type of, I don't know, maybe relative quantification of the peaks. And if you were looking for that half mass, then maybe your peak is like, I don't know, 3 times as big or 4 times as big as you think it is, because actually, yeah.
It's underneath. Yeah. Yeah. No, I haven't seen it.
Okay. And
it could just be like ESI conditions, right? What's happening under ESI conditions, or yeah.
Mm-hmm, mm-hmm.
Oh, sorry, Mohammed, do you have a question?
Yeah. So what was the level of separation in your weeks? And because it's not a knockout approach, how did you deal with that?
Right. So when we silence the polyketide synthase, we get very efficient silencing. So there is a very clear metabolic phenotype. It's not a complete knockdown. So when we report metabolic differences, they're not like there's still tropine and there's still scopolamine in our VIGS lines. But if you look at the chromatogram, and I can show this. I don't think I included that. I definitely could. Here. So if you look at this chromatogram, it's very different from a wild type plant. So this 290.18, that's hyoscyamine. In a wild type chromatogram or in a TRV2 chromatogram, that peak's going to dominate. And these peaks are going to be a lot smaller. So did that answer your question?
Yeah, thank you.
I think I might have a related question, and maybe you just sort of answered it, and or maybe you said it earlier, but any of those 292 derivatives, did you detect those in the wild type, or is that what you just said? Or like, and do you have to be present at such low levels that you need to do a targeted analysis, or do you even see them in like a untargeted metabolomics approach? So
if you are doing untargeted metabolomics on wild type plants, the 292s are there. They're actually quite abundant. Probably like, they're definitely not as abundant as hyoscyamine or scopolamine, but maybe like half the abundance of tropine. The derivatives, so the glycosylations and the decorations and the dipyrrolidine versions of the 292 are there, but I only see them if I concentrate a wild type sample and do deeper profiling.
I see.
So they do exist in wild type plants. They're just comparatively a lot lower abundant than tropane alkaloids.
Got it. Thank you. So there's like a continuum of these, and you basically are adjusting the abundance of the whole continuum when you create the backup. Okay.
Yeah.
Thank you. And great talk, by the way. Other questions for Hannah? Alrighty, well, if there aren't any other questions, then I would like to thank you all for being here today. And I'd like to give one last big thanks to all the speakers for this whole spring session. It's been a really fun session, and I'm really looking forward to seeing you all again in the fall. And please remember, if you or someone you think should speak in the fall, please don't hesitate to nominate them on the website. So I wish everyone an excellent summer, and we'll see you all again in a few months.